Salvete Cristiani, fratres et sororos meum. So, today, what are we talking about? Of course, biology. But we're talking about virology, which is kind of in biology. Today, we're going to talk about what are viruses. Remember, I said that I was going to explain viruses in another video? Well, I did touch up on viruses, last video when I talked about the 8 characteristics of life. But today, we're going to go a little more in depth of what viruses are. So let's get to it. Oh my gosh, I'm trying to pause my video. Gosh. So we're going to do a little recap, a little review from our last video. All right, so what's virology? The study of viruses, right? Virology. So we're going to do a little recap. Remember, I had asked, we asked, are they alive? Why? How come they aren't in the, not the animal kingdom, but the kingdoms of life, 5 kingdoms. Protists, fungi, animals, plants, uh, bacteria. Yeah, remember? So we're going to talk about, are they alive? Let's just recap. Let's look at some reasons why they could be alive. They have genetics. They can cause serious infections. They can reproduce, and they can evolve. But let's look at some living characteristics. A virus does not have a cell. It has no metabolism. It cannot live on its own. And a virus does not react to antibiotics. Now let's go over some of the properties of viruses. So viruses aren't living, or they aren't living, or they're non-living. They're kind of both. They're kind of living, and they're living. I mean, they're kind of not living and living. Of course, they have no cells, like I just mentioned, or any type of cellular component there may be. Move or grow. They cannot reproduce, and the only time they're reproducing is when they're inside of a host cell. Viruses are very, extremely small, smaller than the cells in our body and inside our body. And they're only visible, and like the only time you can see them is under an advanced electron microscope. Other than that, yeah, you're not seeing them. So let's talk about the shape of a virus. Now there's 3 shapes. Your rod shape, then you have your spherical and oval shape, and then you have your tadpole shape. Because viruses can be shaped like a tadpole. It's funny. Like I was in biology class and I was looking because the teacher he put up the shapes. I'm looking like the tadpole looks like some type of robot. It does. All right. I'm gonna try to. I don't know, but I'm gonna try to put pictures inside the video of what the 3 viruses look like. But anyway, yeah, like he was showing me. I'm like that kind of looks like a robot. Okay, so let's discuss the structure of a virus. So a virus all viruses are made up of 2 parts: the protein coat, and the core. Outer protein core is known as the capsid. The inner core of a virus either contains DNA or RNA. And viruses are really nucleoprotein particles. Some viruses that you may see have an extra coat called the envelope. This coat is made up of lipids and proteins. If you look at it like this way, the capsid has the protein. The inner core has the DNA and RNA, and then, like I said, some viruses have an envelope, the extra coat, that contains lipids and proteins. Okay, so viruses they reproduce in 2 ways, and well, the 2 cycles. There's a lytic cycle and there's a lysogenic cycle. We'll go over a little bit of what they are. Okay, so the lytic cycle. After the lytic cycle is complete, the whole cell is destroyed. But for lysogenic, it's not even damaged. After 1 cycle, which takes about 30 minutes to complete of a virus, the virus will have created 300, you know, offsprings. And it only takes 30 minutes. I mean, look at us humans. It takes a year to produce 1 baby. As for the viruses, after 30 minutes, there's 300. So let's say, you get sick, like in the morning, you just wake up, you feel fine, and the virus is just making babies. By the end of the day, you're over here like, yeah, that's what happens. You see? So let's see how the steps of the lactic cycle. So first step, you have attachment, and then the penetration, then the replication, then the assembly, and then the release. That's what happens inside the lytic cycle. Now let's look at lysogenic cycle. In a lysogenic, the virus is attached to the cell, the attachment. And then the virus will inject DNA inside of that cell. Then the DNA, the virus, it forms a circle and it injects it inside the chromosome. It injects itself inside the chromosome. And then the virus DNA, it's replicated. So that means like once it injects it inside the chromosome, it's just gonna start reproducing those type of cells. Because you see, after in the lysogenic cycle, the virus then leaves the cell, and since it changed its DNA, which is the instructions of how to, maintain the cells and whatnot, it's going to just reproduce these virus infected cells, and yeah, that's how you could get sick right there. You see? After that virus injects, itself inside the chromosome, it can then leave the cell. Then, once that occurs, it enters the lytic cycle. Right? I said it's attachment, replication. No, that it's attachment, penetration, replication, and then it's the assembly, then a release. But the main difference is between, you have to remember that lytic, it gets destroyed. The lysogenic, well, it doesn't get damaged or destroyed at all. You see? And now we're going to go over a few examples of some viral diseases. So you have HPV, you have AIDS, you have HIV, herpes, Ebola. There's a lot of them. You have West Nile virus that comes from mosquitoes. So those are just some examples of some viral diseases. Well, that's pretty much it for virology. See, it's biology class, not a virology class, but I thought I'd share something about virology with you guys because that's what we learned, and I guess that was one of our standards were the differences between the lysogenic and the lytic cycle. So yeah, that's just a little something about viruses. It's a little short little video I'm putting out. I might upload another video, probably government. I don't know. But yeah, that's pretty much it for today. Bakit temor? Wala.